Assalamu alaikum dear students. Let's discuss lactic acid bacteria as protein therapeutic agent. Lactic acid bacteria are widely used in the food industry. They are used for the production and preservation of different fermented food items. And they are also generally regarded as safe. This is the technical term GRAS, G-R-A-S, which actually describe those elements, those components which are used as the supplements or the safe, generally regarded as safe elements. Lactic acid bacteria are the part of our indigenous gut microflora, and among them it is the lactic lactobacilli, which is also used as probiotic agent. So these probiotic agents are the ones which are live microorganisms which have the ability to confer health benefits, and they do this by altering the indigenous microflora of the gastrointestinal tract, GIT. So their major uses include treatment of gastrointestinal disorders like lactose intolerance, traveler's diarrhea, antibiotic-associated diarrhea, as well as immunological disorders, for example, Crohn's disease and ulcerative colitis. It is particularly important to note that not only lactic acid bacteria are easy to deliver orally, but they also have the potential of mucosal and systematic immune responses. So they easily elicit these responses. So in the past, lactic acid bacteria, they have been used as an excellent host expression systems for expressing different foreign genes. For example, you can see here that the therapeutic protein, cyst wall protein 2, has been expressed in the Lactococcus lactis, and this was used for targeting the pathogen Giardia lamblia. Then we can also see human interleukin-10, catalase enzyme, and cyanovirin, epidermal growth factor, etc., which have been successfully expressed in Lactococcus lactis, and they were used for targeting different pathogens or pathological conditions or diseases. So what is this Lactococcus lactis? It is a wonderful bacteria or prokaryote to be used for expression systems because it is non-pathogenic. At the same time, it is non-invasive, it is non-colonizing, and it is a gram-positive bacteria which do not form inclusion bodies the way E. coli can. And it can give higher expression of proteins within the host cell, within itself, like it can give a higher expression of the protein which is being expressed in it. And it is also used for direct protein delivery into the host cell, like we can use it for production of protein within the human host rather than going for expression outside the human system, and as a result there won't be any downstream processing steps associated to it. So this is the continuation of the same table, and you can see the therapeutic proteins including tetanus toxin fragment C, beta toxoid, envelope protein, nucleocapsid protein, etc. These are successfully expressed in Lactococcus lactis, and this column actually shows the targeted pathological conditions or pathogens or diseases. Keep in mind there is a distinction between the use of Lactococcus lactis to deliver human or animal therapeutic agent and its use as a live vaccine. When we say that it is being used as a therapeutic agent, this means then it has a biological activity of its own. But when we use Lactococcus lactis as a live vaccine, that means that it will deliver the protein that will elicit the immune response in the host. So we have to clarify this. So under this lactic acid bacteria, I will discuss the 3 proteins which have been expressed. Examples of these 4 proteins we will discuss in detail inshallah ta'ala, and these include interleukin-10, leptin, HIV inhibitor, and let's begin with interleukin-10. For that, we have to consider two pathological conditions, ulcerative colitis and Crohn's disease. These are the diseases, immunological diseases, which affect 1 in 500 to 1,000 person in the developed countries of the world. When we talk about ulcerative colitis, it is associated with excessive type 2 T helper cell cytokines. And those cytokines include interleukin 4 and interleukin 5. Whereas the Crohn's disease is associated with excessive production of type 1 T helper cell cytokine, including TNF alpha and IFN alpha, as well as interleukin 2. So the treatment for both diseases involves lowering the levels of cytokines. When we lower the levels of these cytokines, then these inflammatory diseases settled, or these can be managed. So there are different approaches used for the treatment purposes. The first approach is the administration of antibodies against those cytokines. For example, if the concerned cytokine is TNF alpha, then the administration of antibodies against TNF alpha will be used. The second approach is targeting interleukin 10. Interleukin 10, why interleukin 10? Because it modulates the regulatory T cells. And those regulatory T cells in turn control the inflammatory responses of the intestinal antigens. So that there is this indirect method of controlling or managing those diseases. But the problem is interleukin 10 is not clinically acceptable, because when it is used, that means that it should be injected very frequently, and it can cause rectal enemas. So the solution is using engineered Lactococcus lactis to synthesize and secrete interleukin 10 within the human system. So for that purpose, experiments were performed in the mice to test whether interleukin 10 secreting Lactococcus lactis could be used to treat inflammatory bowel diseases or not. These GIT tract diseases and the immunological diseases, they come under the umbrella of inflammatory bowel diseases, which is abbreviated as IBD. So what they did, they not only took the interleukin 10 deficient mouse models, but they also took a mouse model which was treated with a normal mouse which was treated with 5% dextran sulfate. And this 5% dextran sulfate actually causes inflammation of the intestines. So in both conditions, whether it is a genetically knockout model of interleukin 10 deficient mouse or the one produced with the help of dextran sulfate, the phenotype is ulcerative colitis. Then for both of these models, engineered interleukin 10 producing Lactococcus lactis was given. And it was noticed for the knockout model of interleukin 10 deficient mouse, it prevented the onset of ulcerative colitis, which is a very promising result of its own. And for the second case, in which 5% dextran sulfate was used for generating ulcerative colitis or inflammation of the intestine, there was 50% reduction of the ulcerative colitis. So the results were very promising, at least in case of mice. But the problem lies here is, or the concern is, that the mouse model generated for these IBDs are not identical to human inflammatory bowel diseases. So the question remained before the treatment could be used with human or not. So for this, they came up with, then there was another concern that when we use genetically modified bacteria, for example, this interleukin 10 secreting Lactococcus lactis strain as therapeutic agent, then there is a possibility that this genetically modified bacteria can get released into the environment. If this were to happen, a plasmid carrying the interleukin 10 gene and any plasmid-borne antibiotic-resistant marker gene could be spread to other bacteria in the environment. So this is a very alarming situation of its own. Then scientists took different steps for the safety to prevent this from happening. What they did, they generated a synthetic interleukin 10 gene, and then they constructed this genetic construct. They used the leader peptide fused with the synthetic interleukin human gene, and it was kept under the promoter for thymidylate synthase gene. This thymidylate synthase gene promoter will turn on only in the presence of thymidine or thymine. You can also see the flanking region on both sides of this gene cassette, which is used for the homologous recombination purposes, so that this genetic construct can be used to replace the original thymidylate synthase gene in the Lactococcus lactis chromosomal DNA, fine, with the help of this homologous recombination. So then they checked the results. First of all, they did it by growing it in the laboratory, and they found that the viability of the bacterium declined by many orders of magnitude when it was deprived of thymidine or thymine. But when thymine and thymidine is present, only then it expresses itself. The next thing was this modified bacterium was also used in the pigs. Why pigs was used? Because the pig's digestive tract is similar to that of humans. So when it was tested in pig models, not only that the modified interleukin Lactococcus lactis thrived, but it also produced a sufficient amount of interleukin 10, and that lowered the symptoms of ulcerative colitis. Additionally, it was found that this modified lactolactis was extremely unlikely to acquire a thymidylate synthase gene from other bacteria in the environment. As a result, the scientists were pretty confident that it is safe and it is effective to use in humans. So they established to some extent the safety and efficacy of this approach. Then, as per this chapter and the book of our Glegg, which was published in 2017, there were promising results coming from the phase 2 clinical trials with the interleukin 10 secreting Lactococcus lactis strain, and the patients who received this treatment, they showed significant decrease in the Crohn's disease activity, and only minor adverse events were found in them. Also, the engineered bacteria isolated from the patient's feces was not able to grow without the addition of thymidine. So, in other words, we can say that that engineered Lactococcus lactis did not acquire thymidylate synthase gene from the environment, and that ultimately proved that the containment strategy was effective. So now I have an assignment question for you. You have to go through that clinicaltrial.gov website, and you have to tell me whether this treatment has been approved to date or not. With this, I'm winding up my talk. Thanks for listening.